everyone. Welcome to lecture 40 of BMS 300. Right, we are way beyond the halfway point. All right, so today we'll be wrapping up with the immune system, and we'll move on to the digestive system. Right, my goal is to make sure we finish digestive system before you all go for Thanksgiving break, so that you can impress your family and friends. All right, so with that, let's pick up where we left off yesterday. So we were talking about the lymphatic system, and we discussed about the lymphatic vessels, how they permit a one-way flow, and then we moved on to the primary lymphoid organs. Right, we talked about what's happening in the thymus as far as the maturation and selection of the T lymphocytes is concerned. And then we moved on to talk about the secondary lymphatic organs, and we were discussing about the lymph node, and we'll again pick up at the lymph node where we left off. And the reason for that is there are some things happening in the lymph node which will play a role in our subsequent discussion of the innate immunity and the adaptive immunity. So the innate immune response will activate the adaptive immune response, and that's going to happen in the lymph node, and that's why I think it's important that we really establish those concepts well. So as far as the innate immune response is concerned, it's very non-specific, it's very quick, but the adaptive immune response is a lot slower, and it is highly specific, right? So that's why we take vaccines and so on. And when we consider the adaptive immune response, we will look at two types of immunity. We'll have the humoral and the cell-mediated immunity. Humoral immunity is where you have antibodies, which are proteins released by these immune cells. They will travel through the bloodstream, and then they can attack pathogens. Right? They can bind to pathogens and thereby mark it for phagocytosis. Whereas the cell-mediated immunity is where you have cells going around looking for infected cells and destroying them. Right. So coming back to lymph nodes, we'll just take a quick look at what are the different parts of a lymph node and what happens in these various parts. So here you have the afferent vessels bringing the lymph to the lymph node, and you have an efferent vessel taking the lymph away from the lymph node. And you also notice there are these blood vessels which carry blood to the lymph node and away from the lymph node. Now this outer part of the lymph node is known as a cortex, right? It's called the cortex, and it has these many follicles found all along the cortex. Now in the outer part of the cortex you have naive B cells. So these are B cells which are mature, but they haven't been exposed to antigens, and so they are naive B cells. Whereas in the middle you have the germinal center in the follicles, and that's where you have the B cells dividing and undergoing a clonal expansion. And what we mean by a clonal expansion is that there are multiple copies of those B cells being made, and these are B cells which have been exposed to danger, and now they have been activated, and so there are multiple copies of those B cells being made in the germinal center. Also in the cortex you will have some of these T cells that are in transit, and these T cells will be performing a surveillance role. So you could have T helper cells which are going to activate the B cells, or you could have cytotoxic T cells which will go around killing cells which are infected. Right? So you have cells in T cells in transit which are performing a surveillance function. And when you move to the medulla of the lymph node, you find that there are these plasma B cells. So the plasma B cells are those activated B cells which are capable of producing antibodies. And you also have macrophages in this medulla, and the macrophages will phagocytose any pathogens which come in through the lymph node and ensure that the pathogens aren't being passed on to other lymph nodes or the surrounding tissues. There is also this nebulous region in between known as the paracortex. So it's found between the medulla and the cortex, and here you will find naive T cells and dendritic cells. So I'll tell you how these naive T cells can be activated in the upcoming slides. But a brief note about the dendritic cells is that they are very similar to macrophages in their functions, right? They will also attack pathogens, but most importantly, they will present the fragments of the pathogens to other cells to train them to be able to recognize those fragments. And those pathogen fragments are also known as antigens. And these dendritic cells and macrophages are capable of presenting antigens so that they can train the immune system to recognize those antigens. So there are two major functions of these lymph nodes, and the first one is to clean the lymph, right? And I told you that this lymph node is going to act as a filter through which the lymph flows. And what we mean by cleaning the lymph is that the macrophages in the lymph nodes, they will destroy the microorganisms which enter into the lymph node. And by destroying these microorganisms, we see that the pathogens or microbes can't go to the surrounding tissues or go to the other lymph nodes or even go back into the bloodstream. So it's going to prevent the pathogens from spreading. Now, once a pathogen has been destroyed, those fragments of the pathogens can be used to activate the immune system. And notice that these lymph nodes and also the other lymphatic tissue, they will be found in very strategic locations, like you find them in the tonsils, which are going to be found in proximity to the respiratory passageway, or tonsils found near the oral cavity. And by having these lymphoid organs positioned very strategically, they can encounter the antigens, and it can use those antigens to mount an immune response. And here's another place where you have these lymphoid tissue, like in the Peyer's patches of the small intestines. And we see that some of the cells involved in presenting these antigens are the dendritic cells. They will present the antigens to activate the T cells in the lymph nodes. And so here's a cartoon showing you how an antigen presenting cell is showing a piece of antigen to a naive T cell, and then this naive T cell gets activated and it can recognize the antigen, and if a cell is presenting that antigen, it would kill it, right? So this is one type of T cell. This is the cytotoxic T cell. And we also see how the T helper cells can be activated as well. But for now, I just want to briefly touch upon the spleen, right? So the spleen is also a secondary lymphoid organ, because you will have the T and the B lymphocytes being activated in the spleen. But if you were to do a cross section of the spleen, you understand that it has the red pulp, where there will be a number of red blood cells, and in the red pulp is where you will have the red blood cells being monitored for the level of oxidation of the glycoproteins, which are found on the surface of the red blood cells. And if the level of oxidation is at a certain threshold, the red blood cells would be phagocytosed. And then you have the white pulp, which will resemble the lymphoid nodules, and that's where you'll have the activation of the B and the T lymphocytes taking place. So I mentioned that the abnormal B cells will be phagocytosed in the red pulp, and the iron which is released from the hemoglobin can be stored temporarily in the spleen, and from there it can be sent to the bone marrow for making more red blood cells. Okay, can you still hear me, folks? Yes? All right, great. And because of the white pulp looks like the lymphoid nodules, you will even have an immune response to antigens mounted by the various immune cells found there, like the macrophages, B cells, and the T cells also. So with that, we move on from our discussion of the secondary lymphoid organs, and we will talk about the adaptive and the acquired immunity. So if you were to take the whole blood sample and spin it down, we see that the supernatant contains the plasma, and the pellet down below has the red blood cells and also the buffy coat where you have the white blood cells. And our focus is going to be on the white blood cells, which are found in the buffy coat. So the cells which I will list out below for you, they are playing a role both in the innate and in the adaptive immune response. So the cells we consider will be mast cells, neutrophils, eosinophils, and monocytes, which will differentiate into macrophages and dendritic cells. And we'll even talk about T and B lymphocytes. So we will see how these B lymphocytes can differentiate into these plasma cells, and these plasma cells can produce antibodies which will target specific antigens. Another type of B cell is the memory B cell. The memory B cells, they have a much longer lifetime, and they can convert into these plasma B cells when they can help make more plasma B cells when they encounter an antigen which they have been formerly exposed to. Now when you consider the types of T lymphocytes, we have the T helper cells, also known as the CD4 cells because they express the CD4 protein on their surface, or you could have the cytotoxic T cells, also called as CD8 cells because they express CD8 protein on their surface. And among these 12 different cells I mentioned to you, the ones in the top panel, they all play a role in the innate immune response, whereas the ones in the bottom panel, they'll play a role in the acquired immune response. Yes?
What do the dendrites on the dendritic cells do?
So they play a role in phagocytosis of the microbes. The question was on what was the function of the dendritic cells. They'll also play a role in attacking the pathogens. Yes?
Can plasma cells all be memory B cells, or just some of them?
No, so the B cells can divide into either plasma cells or memory B cells. Yeah, and the plasma cells will produce the antibodies to bind to specific antigens, whereas the memory B cells will just remain in the body, and when they encounter antigen which they are familiar to, they will cause more plasma B cells to be produced. And I'll tell you the few differences between the memory cells and the plasma B cells for the scope of our discussion. For those of you who have taken a more advanced immunology course, you'll see that I'm simplifying things tremendously because we can't go deep because we're going very wide in terms of the material. Yes?
Can you just explain what the innate versus acquired means?
I will. I'll explain in a second, right? So I will explain what is the difference between innate and the acquired immune response. So some of these cells which we just now listed out, they are all old friends. If they aren't familiar to you and they aren't your old friends, here's an opportunity to get familiar with all of them, right? So the first time I mentioned an immune cell to you was back when we discussed the integumentary system, right? So we talked about the different layers of the epidermis, come, let's go, sunbathing, corneum, lucidum, and so on, right? So the stratum spinosum is where you find the Langerhans or the dendritic cells. And then when you go deeper into the from the epidermis into the dermis, we see that the upper part of the dermis is called the papillary layer. That's forming 20% of the integumentary system, and it's made up of areolar connective tissue. But the remaining 80% of the dermis is made up of it's called the reticular layer, and it's made up of the dense regular connective tissue. So as far as the immune cells are concerned, our focus will be on the areolar connective tissue, and here are some of the cells we have previously encountered, right? So we talked about we have heard about mast cells, macrophages, neutrophils, plasma cells, lymphocytes, and so all these cells here. Notice that both cells related to the innate and the adaptive immune response are found here, right? And so we'll see how they play a role. And in brief, let me mention that this innate immune response is what will happen immediately when a pathogen is encountered, and it is not specific, but the acquired immune response will be very specific to the antigens which are found on the pathogen. So just to refresh your memories about the mast cells, notice that they have a similar function to the basophils because they can release histamines and heparin as well. And histamine will play a role in triggering an allergic response, and also it can act as a vasodilator so that more blood can be brought to an injured region so that it can heal up. And keep in mind that more blood is brought there after the blood clot has taken place and blood losses has been prevented. And heparin will be an anticoagulant because when you have more blood coming to a specific region for the sake of wound healing, you don't want the blood to coagulate. So all these cells here, we'll see how they are going to work together if you have a splinter get into your skin. But before we get into the details of the innate immune response, I just want to give you an overview answer for this question: how does the innate immune response trigger the adaptive immune response? So I mentioned previously that the non-specific or the innate immune response is kind of like a grenade which will destroy friend and foe. It's not specific, whereas the specific or adaptive immune response is it's very particular to which type of microbe is attacking, but it tends to be a lot slower. And according to our analogy, it's like a sniper taking a nap, right? And this sniper needs to be woken up when this grenade goes boom, right? And all of the fragments of the pathogens which are scattered by this grenade blasting, they are going to be used by the sniper to mount a very specific immune response. So the cells involved in the innate immune response are shown on the left hand panel, and on the right hand side we see the cells involved in the acquired immune response. So once there has been an innate immune response and these dendritic and macrophages have dendritic cells and macrophages have attacked the pathogen, there will be fragments of these pathogens which are the antigens, right? And these fragments are going to be presented by these antigen presenting cells. And keep in mind that the naive B cells can also act as an antigen presenting cells, not just the dendritic cells and the macrophages. So once they present the antigens on their cell surface, they can activate either a cytotoxic T cell or a helper T cell. And when the antigen is presented on an MHC1 protein, it will activate the cytotoxic T cells. But if the antigen is presented on an MHC2 protein, it will activate the helper T cells. And the reason why when I think of the word presentation, I think about food presentation, right? You could also think about class presentation, but you guys may relate more with food being presented. That's why I used these icons here. So cytotoxic T cells are also known as CD8 cells because they express the CD8 protein on their surface, and these cells will kill any of the cells which present an antigen on MHC1 protein, right? So cytotoxic cells will kill, whereas the T helper cells, also known as the CD4 cells because they express the CD4 protein on the surface, they will help the cell which is presenting the antigen to develop antibodies for that specific antigen. So the CD4 cells can help make plasma B cells which will produce the antibodies, or it can also make memory B cells which will ensure that the antibodies are produced very quickly. So the first time an antigen is encountered, right, it will take around 8 to 14 days for these plasma B cells to be produced, and these plasma B cells will produce antibodies to bind to the antigen. And when an antigen binds to when an antibody binds to the antigen on the surface of a pathogen, it can impair the functioning of the pathogen, or it can also mark that pathogen for phagocytosis, right? And I will tell you there is a specific region on the antibody which the macrophage can bind to, and that's how it can engage in phagocytosis. Now, if the body has already been exposed to a specific type of antigen, there will also be these memory B cells which will have a very long lifetime. It can last for decades, right? And these memory B cells, when they encounter an antigen which they are familiar to, within 4 days, right, it can start permitting more of these plasma B cells to be produced, and it can mount an immune response. And before there are any symptoms noticeable, that pathogen can be dealt with, all because of these memory B cells, right? The details on the difference between the plasma B cells and memory B cells is beyond the scope for our discussion. I can only tell you how they differ functionally, but in terms of structure and the types of proteins being expressed on their surface, it's beyond the scope of our discussion. Yes, what's your question?
So, do dendritic cells and macrophages, are they basically like detectors of an antigen?
Yes, and I'll tell you how they will be brought to a place to detect an antigen in a minute. Yes?
So do CD8 cells and plasma cells both play the role in like killing like foreign?
Yeah, both will play a role in attacking the invading pathogens, but it's through different mechanisms, as I will explain in the upcoming slides. Are we all clear up to this point how the innate immune response will activate the adaptive immune response? Just the overview of how that happens. We'll get into the details in a couple of minutes. Okay, so just one more thing. You've heard this term cellular branch of immunity versus humoral immunity. So the CD8 cells will fall under the branch of the cellular branch, because you have cells going around detecting infected cells and killing them, right? Whereas the humoral branch would be the plasma B cells, because they are releasing antibodies into the bloodstream. So the word humoral is just referring to body fluids, right? And you're already familiar with the word humoral, because when we talk about the 3 types of stimuli to cause the hormones to be released, we talked about neural stimuli, hormonal stimuli, and humoral stimuli, right? So humoral stimuli would be a change in blood phosphate level or calcium level or blood glucose level. All that will be a humoral stimuli, and this humoral branch of immunity is releasing antibodies into the blood plasma. Does that sound good? Make sense? All right. So now let's talk about the innate immune response to a bacterial infection. Right. So here's a cartoon of the epidermis. Below that you have the dermis, and here's a splinter. Ouch! For the sake of sound effects, right? And since you have hit the pain receptors and you've also struck some blood vessels, here's some blood coming out. But notice what has also entered in. You have these bacteria which have entered in because of this splinter piercing down to the dermis. Now the bacteria can cause some damage to the dermis, and that can in turn lead to PAMPs and DAMPs, right? So PAMPs stands for the pattern associated molecular patterns. So this could be the lipopolysaccharides on the surface of the gram-negative bacteria. I already gave you the definition before. And DAMPs could be the substances which are released due to the damage of the tissue. And an example of this would be an increase in ATP concentration in the extracellular fluid because of the cells which have died and released their ATP. And both PAMPs and DAMPs, they can be detected by the immune system, and they are specifically going to activate these mast cells which are found in the what connective tissue? In the what connective tissue are the mast cells found in? Areolar connective tissue, right? So the mast cells will degranulate to release their histamines and the heparin, and the histamines, they are going to play a role in the inflammatory response. And so there were these 4 cardinal signs of inflammation which were described back in the time of gladiators. The physician assisting gladiators or helping gladiators, he found these 4 signs, and we can say that there's also a 5th consequence of inflammation, right? So the 4 things described were heat, redness, swelling, pain, and eventually there will be loss of function as well when there is inflammation. So among the first four signs of inflammation, right, we find that pain is due to the nociceptors being activated, right? When you have a splinter coming in, it can activate the nociceptors and that can lead to pain. Whereas the remaining three, redness, swelling and heat, all of that can be mediated by the action of histamines. And just a side note about the temperature going up, when the temperature increases in a given tissue, that can increase the metabolic rate and that can accelerate the wound healing process. Okay. So there are also going to be cytokines which are released by the damaged tissue, as you can see here, cytokines shown in blue dots, and they can also be chemokines. And these cytokines, what they will do is they will cause the blood vessels to dilate, and here's a cartoon of the dilated blood vessel. And the heparin, which was initially released by the mast cells, they will ensure that the blood flow, which is increased because of the dilated vessels, there won't be any clotting of the blood, right? That's why you need heparin to be released. So let's see what these chemokines do, right? The chemokines which were released by the damaged cells, they're going to bring about chemotaxis, which means cells will be attracted to the injured region, and the very first white blood cell which arrives there will be the neutrophils. So here's the neutrophil, and generally speaking, the neutrophils should just flow right along these injured tissue, but this injured tissue will start expressing these receptors known as selectin receptors, and because of the selectin receptors, the neutrophil will bind to the receptor and then it will start squeezing itself between these endothelial cells, and this squeezing of the neutrophil through the endothelial cell is called diapedesis. And so the neutrophil will arrive at the injured region, and once it encounters the pathogens, it can start shooting out these traps which will trap the pathogen, and it also starts acting like a flamethrower. It releases hydrogen peroxide, free radicals, and basically the damaged cells will explode because of all of these harmful things released by the neutrophils. And even the healthy cells in the vicinity and the bacteria will be killed by the result of the neutrophils. And as a result, even the neutrophils will even die when they release all their contents. So that is why when you have pus formation, it's going to include the dead neutrophils and the cellular debris which was caused by the neutrophil releasing its contents. Okay. Are we clear up to this point? Yes, yes.
How does the heparin not dissolve in the blood clot?
It will prevent the blood clot from happening. Yeah. You want to know the mechanism of how a heparin works beyond the scope of our discussion. We can maybe talk about after class. Okay, so now that the neutrophils have done their job, keep in mind there are some chemokines which are released in this earlier step here, step number 4. Those chemokines are going to recruit other white blood cells, and they're going to recruit the monocytes from the bone marrow, and these monocytes, they will differentiate into macrophages and into dendritic cells, right? And the macrophages and the dendritic cells will arrive at the injured region, and they will engage in phagocytosis, and they will attack and destroy the pathogens. So that is how the innate immune response happens, right? It's not specific at all. It's very quick. It's very destructive. But the next question is, how does this innate immune response lead to an adaptive immune response? Okay. So we find that it's all going to start with an antigen presenting cell presenting an antigen to a naive T cell, right? And so these antigen presenting cells could be either dendritic cells, macrophages, or even naive B lymphocytes. So in this situation, the macrophage is choosing to present the antigen on a MHC2 protein. And because it's expressing it on it's presenting it on an MHC2 protein, this will be attracted to the CD4 cells. And notice that the CD4 cells, they will bind to MHC2 and bring these two cells in close proximity, just like how when you throw the grappling hook from one pirate ship to another ship and it brings those two ships in proximity. Similarly, these two cells are also brought in proximity. And when they're brought in proximity, there will be an interaction between the T cell receptor on the surface of the T cell that can interact with the antigen fragment. And this is the very first step in the adaptive immune response taking place. So you have antigen recognition taking place, and that is the first step in the adaptive immune response. Now there's a second step following this called co-stimulation, where you have interaction between two different proteins, right? So one of those proteins is the B7 protein, and that will be expressed by any professional antigen presenting cell which has seen danger. So if a macrophage or dendritic cell has seen danger, it will express B7, and B7 will interact with CD28, which is found on the T cell. And so this is called co-stimulation. And this co-stimulation will lead to this T cell producing interleukin 2, and this interleukin 2 will engage in autocrine signaling, because notice that interleukin 2 is released by the T cell, and the receptor for the interleukin 2 to bind is also found on the T cell. And so this is going to lead to proliferation or clonal expansion. So there will be multiple clones of this T cell made, right? And so this T cell is no longer naive, because it has had this co-stimulation with a cell which has seen danger. It is now activated. That's why I put sunglasses on it, right? And it's activated to identify this antigen, which is a yellow triangle, right? And so it's the activated T helper cell, and then you have a whole clone army which can activate which can identify this antigen. So now let's see how these activated T cells are going to help the B lymphocytes produce antibodies specific for this antigen. Now the B lymphocytes should have encountered the same antigen, right? And notice that this antigen is going to bind to a receptor on the surface of the B lymphocyte, and this antigen will enter into the B lymphocyte by endocytosis, right? There it goes in, endocytosis, right? And this B lymphocyte will now present the antigen again on a MHC2 protein, and this activated T helper cell, which has CD4 on it, can bind to MHC2 again like a grappling hook, and it brings those two cells in proximity, and the T cell receptor on this activated T helper cell can interact with the antigen, and you have the antigen recognition, first step. And then there will be co-stimulation, because there's the CD40 protein expressed by the B lymphocyte. It will interact with the CD40L on the activated T helper cell, and so there's co-stimulation taking place by CD40 binding with the CD40L. And then the last step will be a paracrine stimulation. Notice that interleukin 4 is produced by this activated T helper cell, but it's called paracrine signaling because the interleukin 4 will bind to the B lymphocyte which presented that antigen. So can you see how it is paracrine signaling? Because it's binding to a cell in its close proximity. And because of this paracrine stimulation, there will be this B lymphocyte can turn into a plasma cell which will make antibodies which are specific for this antigen which was previously encountered by the T helper cell, right? So that's how the plasma cells are made. Yes?
So is it co-stimulation that activates the T cell?
It is all three. It is a co-stimulation which will activate the T cell to in turn activate the B cell to produce the antibodies. Yes. Oh wait, what? Sorry. Wait, what's your question?
Oh, I was just clarifying. It's the B lymphocyte that then becomes the plasma cell?
B cell becomes the plasma cell. Yeah. And what is your question?
Is the T cell activated? Is this adaptive immunity?
Yes, this is adaptive immunity.
So is the T cell activated in both adaptive and innate immunity, or just in innate?
Only in adaptive immunity, right? So it's the T cell is activated as a result of the innate immune response when the antigen presentation takes place. Okay, so these antibodies which are formed, right, they can recognize this specific antigen, right? So here's a pathogen showing expressing the same antigen, and these antibodies will bind to the antigen, and thereby it will impair the functioning of this pathogen, and it can also mark it for phagocytosis, right? So there will be a specific site on this antigen where the macrophages can recognize it, right? So let's take a closer look at the antibody structure. So this antibody cartoon is expressed in as a Y-shaped protein, but keep in mind it has got 4 subunits, right? It's got 2 light chains shown here in light blue, and there are 2 heavy chains shown in purple, and all of these are held together by disulfide bonds. Now this region down here is known as the constant region, and each of the arms of the Y, they are called variable regions, and because that is the place where the antigen would bind. So for different antibodies, depending on what antigen they are going to bind, this antigen binding site in the variable region would be unique. And notice here in this constant region you have the site for the macrophages to bind, and that's how it can once an antibody binds to a pathogen, it can mark it for phagocytosis because of this site where macrophages can identify it. And I hope you all realize that there are 5 different classes of the antibodies, and so we will not have time to talk about it, right? This is just a primer for you. If you're interested, please take a more advanced immunology course. So now we've only talked about the cytotoxic T helper cells. We've talked about the T helper cells. We need to see how the cytotoxic T cells are activated. And if you understood how the T helper cells are activated, this should be pretty straightforward. There are only a couple of differences. So we start with the antigen presenting cell presenting an antigen to the naive T cell, and the antigen presenting cells could be dendritic cells, macrophages, or naive B cells. But the major difference here is that the antigen is presented on a MHC1 protein, right? That's the first difference. And because MHC1 can bind to CD8, we see that a CD8 cell is now come in proximity to this antigen presenting cell. Again, notice CD8 protein is acting like a grappling hook, bringing those 2 cells in close proximity. Then there will be this T cell receptor interacting with the antigen, where you have antigen recognition, and then there will be co-stimulation between the 2 cells because B7 interacts with CD28. B7 is on the antigen presenting cell, and CD28 is expressed by the naive T cell. And interleukin 2 is produced by the naive T cell, which binds to the same cell which produced it, and that can lead to proliferation and clonal expansion. So we find that this CD8 cell is now capable of recognizing this antigen, and notice that I've given it a little sword because it's capable of releasing perforins which can poke holes in the infected cells. And here you have this entire army of cytotoxic T cells which can recognize cells expressing this antigen. Okay, so now let's move on to discuss about the innate response to viral infection, right? So here's the lung epithelia, and we will consider the example of H1N1 virus, and this is the apical surface shown here, and you have the basal layer seen here. And the term H and N actually have a meaning, right? So H stands for hemagglutinin, which is required by the virus for entry into the cell, whereas N stands for neuraminidase, which is required to exit the infected cell. So here's the virus binding to a receptor, right, and then it's going to enter into the cell. And once the virus gets in, it's going to hijack the machinery inside this infected cell to start reproducing and making multiple copies of the virus. And then once it's multiplied, it can leave the cell by cutting ties using neuraminidase, both figuratively and literally, right? So the virus can escape. So now this cell which is infected is at a point of no return, right? So it's expressing a fragment of the virus on MHC1 saying, please come and kill me, I'm infected, we can't move on, right? So this MHC1 fragment will be recognized by cytotoxic T cells, which were activated as mentioned in the previous slide, and they can kill this infected cell. Now this infected cell can do one more thing to prevent the spread of the virus, and that is produce interferon type 1, which will bind to receptors on neighboring cells, and it tells the neighboring cells to shut the doors, close the windows until the storm is past. So literally what's happening is it tells it to decrease the rate of endocytosis so that the virus can't get in. It's also going to decrease the rate of exocytosis and also decrease transcription and translation so that the virus can't keep multiplying inside. So with that, we can move on to the functions of the lymphatic system. Did you guys get it? Endocytosis, exocytosis, transcription and translation are all decreased by interferon binding to these neighboring cells. Alright, so now let's talk about the functions of the lymphatic system. We see that it will produce, maintain and distribute lymphocytes to other lymphoid cells, and how that exactly happens, that's what we talked about for the last half an hour or so. We also see that excess fluid from the tissues is returned back into the bloodstream through the lymphatic system, and I specifically told you what are the two ducts which bring the lymphatic fluid to the subclavian veins on the left and the right side. And we find that the lymphatic system will also help regulate the composition of the interstitial fluid by transporting hormones, nutrients, wastes from the tissues of origin to the general circulation. So it helps to bring substances back to the general circulation. So let's quickly summarize what we discussed about the lymphatic system, right? So we see that the components of the lymphatic system include the vessels, the primary and the secondary lymphatic organs, and the innate immunity will deal with the immediate threat from pathogens, but it can also activate the adaptive immunity by presenting antigens on the cell surface. Right? Now, humoral immunity is mediated by the antigens presented on MHC2 proteins, and that will activate B lymphocytes, whereas the cellular immunity is mediated by antigens presented on MHC1, and that can activate the T cells, the cytotoxic T cells, and finally the interferons will mediate the innate response to viral infection, right? So with that, I want to very quickly talk about the